Thank you, Asunta.
Thank you, Andrea.
And hi, everybody. I'm changing completely the subject. Now I'm talking genetics.
Let's go to the plan.
I'm going to present our work in the genomic screening for tolerance of coconut population differentially exposed to coconut lethal yellowing in Ghana using GWAS. So, coconut has been acknowledged as a tree of life because of this enormous number of uses that are alimentary or not alimentary, but also because it's a source of income for millions of smallholder farmers all over the world. Coconut diversity is very, very wide, and that is a reflection of these two origins, one in the Indic Ocean and the other in the Pacific Ocean. So from where it was spread all over the intertropical zone, all over the world. But this large diversity sometimes is very hard to explore, to use in coconut genetics diversity. And that is because of the different biological characteristics of coconut, such as the large seeds that it has, that it requires large surfaces to grow and maintain. It has a long generation time, low prolificacy. It's a multi-purpose crop, and because there's a lot of threats and emerging diseases, and also because of climate change. So between these threats, maybe the most important one for coconut cultivation is the lethal yellowing, associated with the phytoplasma. So up to now, there is no known resistance genes known to lethal yellowing. And one of the interesting observation is that when an epidemic occurs, so more or less 5 to 10% of the plants can survive or are survivors. So maybe this plant might have some kind of resistance or tolerance to the phytoplasm. So in order to explore this possibility, the subtask 3.3.3 from the Tropicsafe wanted to find putative resistant individuals among the survival within a generally susceptible population, the West African toad. Also, the idea was to identify genomic regions associated with the plant survival through association genetic studies, using plants that are in places affected with lethal yellowing and others that are in unaffected places. So the first thing what we did was a screening in the south of Ghana, here, where 6 different sites were visited in order to look for survivors. So at the end, 40 survivors were found here in red in the different places and collected. And as controls, plants that are were in places where the disease wasn't yet arrived were taken also as control. Here you can see them as blue. As a comparison, also 5 individuals of reference individuals were collected. So as you could say in the presentation, the way of taking the tissue is like we're taking the sawdust from each one of the plants. This sample was then taken to Montpellier, where DNA extraction was carried out, and also the GWAS library construction was done. Then those libraries were sent to sequencing by Illumina per end. So when we had our raw data from this Illumina sequencing. We use, for the genotyping, we use our high non-tall reference genome, a genome that we constructed in collaboration with Dr. Yao Dongyang from Criccatas. We constructed this genome that at the end we had around 2.06 gigabytes of genome assembled in 16 chromosomes. We use this genome and the raw data to do the SNP calling. Then when we have this SNP calling, we have applied some pre-filters in quality, mapping quality, mean coverage, and missing data to eliminate bad markers. We also did the same kind of filtering with the individuals by quality, by coverage. We eliminated the ones that were not high enough. Then we carry out an imputation data of missing data to complete our data set. We did also another individual filtering where all the individuals that got out of the heterozygosity too strong or too high were eliminated. And finally, we only keep the biallelic markers complaining with the high wire equilibrium with a minor allele frequency greater than 0.01. And at the end, we kept two data sets, one with only SNPs and one another with SNP and InDels. So for this analysis, we didn't carry a standard GWAS analysis. We did what is called a case control GWAS. It's what is done to identify human diseases. So we took these NOR data sets and we used them to acknowledge, to understand the population structure using a principal component analysis. And we also use this information, these markers, to construct a relationship matrix between individuals, a kinship matrix. So we use this data, we apply this data in different genetic models, 10 different genetic models used with one software and two different error packages. And with this, we were able to find associated region with a region associated with the survival. We carry out a haplotype analysis to see what alleles were present in those markers. So as our results, we got around 20 individuals per category, that is controls and survivors, and for each one of the different sites visited. That makes around 119 control and 131 survivors. At the end, we were able to analyze 11,400 SNP and 12,600 SNP and InDels. When we check the population structure of our data, we saw that most of the variation is explained by the first percentile component, and that is the one that separates the West African tall that is used or grown in the most of the on the sites visited from the other individuals and the hybrids that are present in one of the sites. The other principal components, they don't show any explicit population structure. One similar result with the relationship between the samples, we see here that the hybrids that are present in one of the sites are, well, yeah, we can see it there in this part of the feature, the picture, while the West African plants are, well, the relationships are not that strong as it's in here. So for the GWAS analysis, we found an association in chromosome 8, an association with the survival phenotype in chromosome 8 using the model Blinker, the model farm CPU, the general linear model, also with super, and a logistical regression with Walt. And we almost found it, it's just below the significance level with the first regression. If we take a look at the same, and we didn't find it with the mixed linear models. So if we look at the results with when we use the SNP and InDels together, we see practically the same result. It's just that with Blink, we found a little region in chromosome 7 that is also could be also associated, but it was only found with this analysis, while the others are exactly the same. We have the same markers, the same region associated with the same genetic models and statistical models. So with this, we can say that we have a significant association on the chromosome 8 to the phenotype, to the survival of the plants. When we take a look of this region, of these markers, using an haplotype association analysis, we see that all the markers that are considered significant here are also considered as an haplotype block. They are inherited in block. So when we take a look at the different alleles that we find for these markers, we see that the combination TGGA, it's significantly more common in the survivors than in the controls. And on the contrary, the complemental alleles, the CATG, are less present in the survivor compared with controls. So that would mean that this combination is the one that is linked to this survival. So we use this information then to observe the alleles present on the different plants that we have on the field, on the analysis. And we selected the ones where we didn't have any missing data and that had the combination that is more present in survivors associated with the survival. So at the end, we selected 24 plants that might be putatively resistant or at least tolerant to the phytoplasm. So in conclusion, we have found a putative tolerance resistant region on the long arm of chromosome 8 with 4 linked, highly linked markers were found significantly associated with the survival in the West African tall. Then with an haplotype analysis, we found a specific haplotype to be more frequently within survivors than within controls. Also that we found 24 survivor plants that are bearing these alleles, associated alleles, and maybe that we can think that they are considering tolerant or resistance, and maybe those plants can be used as donors on future improvement programs. Also that the markers identified could be used to screen even more plants, survivors in the fields. And one interesting thing is that we could apply the same approach to different local population or in different countries and with other coconut varieties, maybe to find other putative resistant individuals. To find putative resistant regions associated with the tolerance or the resistance. So I want to thank my colleagues in Surat, Stephanie and Fabien, and also our collaborators in the CRICATAS, especially Dr. Yao Dongyang. So Thank you.